The next talk is by Professor Alan Jasanoff. Alan is an associate professor at MIT with appointments in Department of Biological Engineering and also in the Department of Brain and Cognitive Sciences, and he's also a member of the McGovern Institute. The focus of Alan's work is on pushing the boundaries of fMRI technology, that's functional magnetic resonance imaging technology, or brain scanning. And Alan's topic today is dissecting the brain. So Professor Jasanoff.
Okay, well, thank you very much, Jim, and thanks to everybody here. It's a great audience, and I promise you there are going to be no knives. MIT's motto, Mens et Manus, is a great metaphor for the history of neuroscience and for neuroscience research today. And that's because when you think about the greatest advances in how we've come to understand the brain or the stand-in of the mind, they've often come through technology, the more applied side of what we here at MIT and throughout the world do, the manus side. And that's really no more true in any branch of science than in neuroscience, which actually grew out of advances in two disparate fields. And famously, the development of the modern microscope in the late 19th century was what first allowed people to see things at enough detail to actually detect the cellular structure of the nervous system. And that came out largely out of advances in physics and specifically new ways to construct optics with precision. Before these developments, microscopic images looked like this, and imagine trying to see the finest details of a cellular structure when every boundary looks blurred. The second area of research that led to breakthroughs in neuroscience around the end of the 19th century were advances in chemistry, and specifically in the technology for labeling and staining neural structures in the nervous system. The most famous example is the one that's depicted here. It's the so-called black reaction, pioneered by Camillo Golgi in the 1870s, that involves the formation of something called silver chromate salt, specifically in individual cells. So using this chemical reaction, individual cells could be labeled in this very pronounced fashion with black dye extending throughout the fine little processes. And in conjunction with the advances in the optical microscope, this allowed people to see the details of cells, their processes, axons, dendrites, and synapses, the connections between cells. And it was largely these new observations led in large part by the Spanish anatomist Ramon y Cajal, that led to our modern conception of the nervous system as a network of cells, neurons, organized in networks with opposing cells touching each other, communicating information, but with the neuron as the essential element of the function of the nervous system. And that replaced largely this alternative theory called reticular theory, which held that the nervous system was basically a sort of a connected network of sort of almost like canals or reticulae. Ironically, that was actually a view held by Golgi, the person whose chemical reaction actually allowed neurons to be seen for the first time. So the cellular basis of the nervous system was, the discovery of the cellular basis of the nervous system was a dramatic breakthrough, and one that basically set neuroscience along a reductionist path that's been enormously successful and that's taken us to where we are. It's taken us to a bit of a crisis, though. And the reason for that is the question of scale. So neurons are very small, and brains are comparatively very big. The human brain has over 10 billion neurons. Each neuron connects to on the order of 1,000 other neurons. And we think that in order to understand the function of the brain, we need to understand a lot more about what these things, how these cells are communicating with each other than we can here. Just to put that in perspective, since it's Tech Day, let's have an MIT illusion. If you think of Kresge Auditorium as a neuron, well, the brain compared with that is basically most of the Northeast. So if you imagine the complexity of Kresge here, and let's omit the complexity of the brains that are in each one of us, but imagine the complexity of Kresge Auditorium and that level of detail basically being needing to recognize that throughout a structure that is 10,000 times bigger. And now, unfortunately, although Google Maps can see the world from above, we can't do that to the brain. The brain is three-dimensional. It's opaque, and it's hidden in an even more opaque skull with skin and stuff over it. But this is where another great example of technology forwarding our field comes into play, and that is through the development of modern brain imaging techniques. And you've already heard about their application today to study patterns of neural activity. The most famous example of brain imaging, or the most successful example, is the use of something called magnetic resonance imaging, MRI, which came out of advances in the middle of the 20th century in how to understand nuclear physics. So we can use those advances to peer into the brain, to dissect it without a knife, as it were, and to see basically every corner of, in this case, a mouse's brain by panning through it. But what turns out to be a little bit harder is to decipher the meaning of the activity patterns that make this brain work. So first, how do we actually see anything about how the brain works now that we have its structure? The answer is by using something called functional brain imaging, or functional magnetic resonance imaging, that's abbreviated fMRI here. That's the technique that allows us to make images like these and to be different. I'm showing you rat brains with functional MRI, but of course human fMRI is the most famous technique. What we're looking at here is something called a coronal section through a mouse's brain, and the colored pixels are areas of the brain that lit up or that changed their signal when a stimulus was given to this animal. In this case, it was a somatosensory stimulus. And what you can see over here is, in red, the time course, the signal over time of the MRI signal in this region over here as the stimulus is being delivered in that line during this period here. In gray is the underlying electrical activity. So in other words, if one went in invasively and recorded from the actual cells what they're doing during the stimulation, they are signaling electrically according to this sign over here. Now, this signal over here. So it turns out that fMRI is a tremendously powerful technique for studying the brain, for seeing where in the brain things change when stimuli happen, when tasks are performed. But it's actually very limited for analysis of neural mechanisms in particular, and that's because it is still very remote from the fundamental units of neural processing, those cells that Ramon y Cajal and Camilo Golgi were studying. And the reason for that is because fMRI is based on an indirect coupling of neural activity to changes in blood flow. What we actually see when we do this imaging are changes in blood flow that are induced by neural activity. This panel over here shows a network of blood vessels that spans a corresponding network of neurons in a small part of the brain, roughly corresponding to this bit over here. You can see that they sort of embrace each other, or they could embrace each other if they were on the same slide. But on the other hand, the blood vessels are further separated from each other, and they can't tell which neuron is signaling to them when they give rise to these signals like this. And in particular, just to give a very simple example, neurons come in various different flavors. Some of them are excitatory, some of them are inhibitory. They tend to shut down activity in other cells. And the balance between excitation and inhibition in green and red here can't possibly be distinguished by these networks of blood vessels here. So how can we do better than that? That is the challenge that my lab has taken on, many other labs as well. Specifically, we're trying to mediate between the non-invasiveness but relatively but whole brain coverage of this fMRI style of technique and this precision of techniques like electrophysiology that actually measure the activity of individual cells. And the specific direction that we've taken is to try to do, in a sense, what chemical staining did for neuroscience in the 19th century, and that is to add this chemical dimension to transduce neural signals into imaging signals that we can detect that actually arise from the molecular level. And we do that using a technique called molecular imaging. The basic idea is to generate the MRI equivalent of dyes that respond to neural activity and that can be detected by MRI. They're called MRI contrast agents, and we build MRI contrast agents that sense neuronal activity. So what are MRI contrast agents? They're a type of molecule. They're actually very, very different from colored dyes. Some of them are colored, but it doesn't make any difference once they're in an MRI scanner. They're chemicals exemplified by this one here. So many of you may have had MRI scans. Many MRI scans, in turn, are done with contrast agents that light up or darken parts of your body when they're injected into you. And that's shown over here in an assay plate here. So you can see brightening or darkening of signals that have gotten these MRI contrast agents in them. And of course, what's neat about these things is that you can introduce them into humans or animals and see some of the same effects. So these are pictures, MRI scans, of a rat that had the misfortune of getting a contrast agent injected into its eye. Well, it could still see after this, but more to the point, we could see parts of its visual system. So you can see the eye lit up with the contrast agent, and you can actually also see bits of the brain that are connected to the eye, in this case, lit up with the contrast agent. Those are areas called the thalamus and the superior colliculus. So contrast agents come in all shapes and forms. They range from small molecules, like the one shown a second ago, to large molecules like this. I hope you appreciate the sort of Star Wars effect here. This is a nanoparticle. Nanoparticles turn out to be really potent types of MRI contrast agent. And our mission in working with all of these things is to sensitize them to the molecular features of neural activity. And some obvious examples include detecting neural signaling with molecules inside cells, as well as with neurotransmitters, the chemicals that signal between neurons. So a specific example of what we've done to achieve this involves engineering a contrast agent here that has a sort of a magnetic component in the middle that makes it a contrast agent. We engineered this thing to stick to a neurotransmitter and change its properties as a result. So we targeted, we've developed MRI contrast agents to target multiple different aspects of neural activity. I'm only going to tell you about one example because it's the example that we've taken the furthest into the actual brain, all the way from the chemistry to the beginnings of biology. And that is a contrast agent that detects the neurotransmitter dopamine. Some of you know that dopamine is the neurotransmitter most closely associated with certain types of learning, like Skinner box learning, where rats press levers to get reward. You heard a little bit about that kind of paradigm from Drazen this morning. And dopamine is also the neurotransmitter that goes wrong, so to speak, in Parkinson's disease. So we engineered a protein to detect dopamine. We took a protein that is naturally an MRI contrast agent, and we started dumping chemicals on it. We found that by dumping the right chemicals on it, we could make it stop being a contrast agent. So it's very bright without anything, but then when we add ligands to it, small molecules, it becomes darker. So the question is, could we actually make this very specific to dopamine? And we did that using a protein engineering technique called directed evolution, where we make a bunch of different mutations in the proteins, and then we use an assay, a high-throughput assay, to find the variants that do best. And it's a little bit like natural evolution, or like what people think is natural evolution, because it involves making mutations and then performing, in a sense, a selection on them, a natural selection, in this case, an unnatural selection. So we used that procedure to build up affinity for dopamine and lose affinity for competitor molecules, and it was a new approach to generating MRI sensors, and then we started applying it in actual animals. And so we didn't go straight to humans. How many of you, by the way, would volunteer to be our first subject? Okay. All right, well, we got one. Okay, so anyway, see me afterwards. All right, anyway, so the rat, we didn't ask the rat, but so we used rats like this, and we did a very, very simple experiment. You can see it's crude in various ways, and actually when I'm done with this, I think my one volunteer is going to retract his enthusiasm. So this is a rat that's being stimulated with an electrode that's been implanted in a pleasure center, an area of the brain that reinforces its behavior, and the rats continue to press the lever ad nauseum, or at least ad infinitum, over and over again in order to get this electrical stimulation. And so we know that this engages the dopaminergic system. It engages dopamine release. And so we wanted to see, could we actually obtain a functional MRI map of this particular neurotransmitter during the kind of stimulation that was keeping this rat going here. And so we implanted rats with electrodes. We inject our contrast agent into a region of the brain that is important for this. Okay, volunteer, you still want to do this? Yeah. Okay, all right, yes, all right. So you can see we can't see the whole brain yet. We're doing injections. That's because we can't solve all of our problems at one single moment. But the basic idea is to infuse the contrast agent, give the stimulus, and see if we can make a functional map. And so we start out by looking at a specific place in the brain. Can we see signal changes when we give the stimulation? And the answer is yes. We can see little dips in the signal every time we give a stimulus. This is the first evidence of being able to do this kind of functional imaging at a molecular level. Now, the first question you might have is, well, how do you know that that wasn't just the rat twitching? Well, the answer is the rat was paralyzed, but we have a bunch of other controls to make sure that this really is our imaging agent that's doing this. The most, actually, I think in a sense, elegant control is that we took a version of our contrast agent that's inactive, that's the same magnetically, but that doesn't bind dopamine, and we did the same experiment. We could see that while we got the signal changes in the case of our sensor, in light green here is the control, and you can barely see any change. In fact, that little ripple is some sort of artifact, and it's gone away as we've done more processing of the data. We did a second control where we measured the dopamine from using a second technique, and we could see that we could find direct evidence that our sensor is actually directly interacting with it. And I won't go into more detail into it, but it adds up to real evidence that this dopamine imaging technique is really working. And what's, of course, the really neat thing about this for us is that we can actually develop then a map of the dopamine concentration for the first time using this imaging agent. So these are now 3 slices through the brain of a rat that was treated with this imaging agent with the stimuli that were keeping it lever pressing, as I showed you before. And we can see this pattern of orange and red. Now, the pattern of orange and red looks just like the fMRI maps that you guys saw in some of the other talks today, but it's different. It means something at a molecular level. It means that dopamine is being released in those places, and we can even quantify its concentration here. And that has to do with the chemical properties of the agent that we measured in very carefully outside of the rat before. So we've got this map here. We can measure error bars on each one of the pixels and show that these are really real signal differences. We can see in particular a peak signal in an area called the nucleus accumbens core. So this really provides some evidence that the molecular functional imaging can really be performed and start giving us information. In fact, we have dynamic information as well. This is sped up by a lot, but it's showing you the rise and fall of these patterns of dopamine that we're able to detect in this way. So dopamine is only a small part of the brain. Dopaminergic cells are an element of the signaling in a very important set of behaviors, but they interact with a huge set of other elements schematically shown here, and we want to see them all. So the larger mission of my lab is to perform, in fact, a functional dissection using molecular techniques similar to what I've just shown you of the different cell types and interactions. And MRI right now is our best way to do this because it allows us to combine this molecular specificity with relative non-invasiveness one day in humans. We've got a whole set of other imaging agents that detect aspects of neural activity, and I won't go into them in detail, but many of them range from proteins that can be genetically encoded, which means they can be targeted to specific types of cells in the brain in animals, to small molecules that actually might one day be applicable in humans because they're a lot more like ordinary drugs or like actually, in a sense, ordinary contrast agents that you might have had in a medical exam. So last, let me just say that MIT has been a great place to do this research because we have a tremendously broad neuroscience and neurotechnology community. It's one where research in neurotechnology development that spans all the way from chemistry to molecular biology up to actual brains and even people can actually occur. And with that, I'd like to thank MIT and thank you for your attention.